Bismillahirrahmanirrahim. Assalamu alaikum and welcome to Sindh Biotechnology Association. In today's lecture, we are going to discuss on ELISA, the enzyme-linked immunosorbent assay. This assay is used as solid phase type of enzyme immunoassay to detect the presence of ligand. ELISA has been used as diagnostic tool in medicine, plant pathology and biotechnology, as well as it is helpful in quality control check in various industries. ELISA is a plate-based assay technique designed for detecting and quantifying soluble substances such as peptides, proteins, antibodies, and hormones. ELISA is one of the most common method used for the detection for antibodies, proteins, peptides and biomolecules. What is immunoassay? A major question come here. The term immunoassay is combined of two words, immuno and assay. Immuno mean practically antigen antibody reaction and assay mean determination of purity of substance or the amount of any constituent of a mixture. So why it is known as ELISA, ELISA, enzyme linked immunosorbent assay. Antigen antibody of interest is absorbed onto plastic surface that is sorbent. Antigen is recognized by specific antibody that is immuno. This antibody recognized by second antibody, this is also known as immuno, which has enzyme attached that is enzyme linked. The substrate react with enzyme to produce product usually colored. History of ELISA. Radioimmunoassay was first described in scientific paper of Rosalind Susan Yellow and Solomon Berson that was published in 1960. In 1971, from University of Stockholm in Sweden, Peter Palman and Eva Engevall they published papers that synthesize his knowledge into method to perform ELISA. The main and important component of immunoassay are antibody, that is antiserum, antigen, and labeling materials. Now let's see what is antibody or antiserum. Antibody are the proteins that are produced by the immune system which help defend against the antigens. This is the symbol, the picture in purple color. This is the symbol of antibody. It has variable regions that are thought to be the place for recognition and binding with antigen. Then we have antigen. Any molecule that induces production of antibodies when introduced in the body is called antigen. Anything foreign to immune system, that is bacteria or virus or pollen, that is antigen, and this triangle is denoted as a symbol for antigens. Labeling material. In immunoassay, it is necessary to use any marker to know the antigen antibody binding. For such purpose, we label either antigen or antibody with some material that do not interfere with binding. The example is horseradish peroxidase enzyme and substrate we use trimethylbenzidine. This is ELISA kit where we add antibodies, antigens and uh, enzymes, and this is ELISA reader from where we get the readings. The basic principle of ELISA, it uses an enzyme to detect the binding of antigen and antibody. The enzyme convert a colorless substrate chromogen to colored product, indicating the presence of antigen antibody binding. An ELISA can be used to detect either the presence of antigen or antibodies in the sample depending how the test is designed. ELISA was developed in 1970 and become rapidly accepted. In most of the simple form of ELISA, the antigen from the sample to be tested are attached to the surface, then the matching antibody is applied over the surface so that it can bind the antigen. The antibody is linked to enzyme and then any unbound antibodies are removed. In the final step, a substance containing enzyme substrate is added. If there was a binding, the subsequent reaction produce a detectable signal, mostly a color change. There are two types of ELISA, a quantitative ELISA and a qualitative ELISA. In qualitative ELISA, it determines the antigen or antibody is present or absent. In quantitative, it determines the quantity of antibody, the titer, the highest dilution of the specimen, usually serum, which gives positive reaction in the test. So these are the some basic steps which are used in ELISA. In number one, we load the sample in a triplicate into wells of the microplate strips and incubate it for 5 minutes, and after that we rinse it. In step 2, we add primary antibodies to all the well and incubate it again for 5 minutes, and again after that we rinse it. In step 3, we add enzyme linked secondary antibody to all wells and incubate again it for 5 minutes, and then again we rinse it. At the last step, we add enzyme substrate to all the well and incubate it for 5 minutes. These are the basic steps which we used in ELISA. The advantages of ELISA. Reagents are relatively cheap and have long shelf life. ELISA is highly specific and sensitive. There is no radiation that cause hazard during labeling or disposal of waste. It is very easy to perform and the procedure is quick. Equipment is inexpensive and widely available. ELISA can be used to variety to detect variety of infections. The disadvantages of ELISA. Measurement of enzyme activity can be more complex than the measurement of activity of some type of radioisotopes. Enzymes activity may be affected by plasma constituent. Kits are commercially available but not cheap. Very specific to a particular antigen that won't recognize any other antigen. False or negative possible, especially with mutated altered antigen. Types of ELISA: competitive ELISA, non competitive ELISA, sandwich ELISA, which is further divided into direct ELISA or indirect ELISA. The main common steps which are in all those types are collection and processing of serum, coating of wells with antibodies or antigens, then washing, incubating it with test sample, then again washing, incubation with enzyme conjugated antibodies, then again washing, color development by adding chromogenic substrate, and then stopping the color development, and in the last we read the results. Here what we do, first step is coating. Control antigen is absorbed onto well in ELISA plate in coating buffer. Remove liquid and wash the plate. Then blocking. A buffer containing unrelated protein is used to block free sites in the wells. Remove liquid and wash the plate. Then prepare the sample, that is unknown antigen and detection antibody mix. Add test sample to mix all the well. Again remove the liquid and wash the plate. Then detection antibody. Add enzyme conjugated secondary detection antibodies. Again remove the liquid and wash the plate. And then readout. Substrate is catalyzed by enzyme to generate colored readout. In this example, the antigen concentration in the sample was low. The antibodies bound the control antigen that had been absorbed to the plate in the coating step. Application of ELISA. ELISA is used to detect viral contamination like HIV, hepatitis C, or other hepatitis. It also used to detect the hormone level. It helps in the detection of various infections like dengue, human papillomavirus. It also helps us to find specific disease factors like clotting factor, Lyme disease. It helps in the detection of drugs from blood, narcotics, antipsychotics, antidepressants. Help to find allergens in food like egg protein, milk protein, residues in food, antibiotics. It helps to find toxin in foods, also help in other tests like pregnancy test, preclinical screening. That's all for today's lecture. Thank you so much for your patience.